that kind of little distinction there, think of the book of Romans, right? The book of Romans was written to the church in Rome, right? But it was written for that church and was written for us as well. So that's kind of the distinction that they bring out. And N.T. Wright does it specifically when he thinks about what was the religious life of the Israelites at the time of what he refers to as the Second Temple Jewish people. The first temple was destroyed, the second temple was built, and at the time of Jesus, that second temple was the temple in which the Jewish people worshiped. Okay, and so he uses his research with regard to the Second Temple Jewish people to interpret a variety of scriptures. Mostly he uses it to interpret what was happening in the letters in the New Testament, but he's also done it for the Psalms. Okay, and so in interpreting the Psalms, he says if the psalmists project a point of contact between God and men and women in terms of three things: in terms of time, in terms of space, and in terms of matter. And so in his monograph, he has one chapter with regard to time, and he has one chapter with regard to space, and another chapter with regard to matter. We're going to kind of focus on matter. Okay? But to kind of get us to that point, he talks about time. He says the Psalms invite us to stand at the intersection between God's time and our time. And if you go through the Psalms, you'll see comparisons between God's time and our time. So that was the first subject that Dr. Wright looked at. The next one was space, that the Psalms present an intersection between God's space and our space. And in particular, he refers to the Second Temple, that the Israelites at that time saw the temple as specifically the location on earth that God dwelled. That started with the tabernacle. You learn about the tabernacle in Exodus, but then the temple was constructed as a more permanent type of tabernacle, the first temple, and then the second temple as well. So for the Israelites, the temple considered God as God's resting place. That's where God was located. And you see in the Psalms references to that. References to the temple, references to Jerusalem, references to Zion as the place where God dwelled. So the Psalms speak of space. Okay. They also speak of matter. And his fifth chapter is entitled, All the Trees of the Forest Sing for Joy. And he says that the Psalms celebrate, in fact, they positively relish the sheer physicality of creation. So let me read just a little section on that, because that's really what I want us to think about a little bit today, this idea of the celebration of the physicality of matter. He says that the Psalms celebrate, in fact, they positively relish the sheer physicality of nature, its stuff and its substance, its seed times and harvests, the winds and the rocks, the days and the nights, and I'm going to kind of add to that the atoms and the molecules, because that's really what we're going to try and think about today. Okay, and so then he comes to the point where he says, the heavens are telling the glory of God, and the firmament proclaims his handiwork. Day to day utters forth speech, and night to night declares knowledge. The Psalms rejoice in this idea of the physicality of nature. And he refers to it in his chapter as matter. Okay, for, as he says, for lack of a better word. Okay, so we're going to look at celebrate the physicality of matter, and we're going to do it with two things. One is I'm going to use my favorite figure. Okay, this is my favorite figure. All right, and you all love that figure because you look at it and instinctively you know exactly what's going on in this favorite figure of mine, right? But what the figure does is it illustrates what's called the Kelvin cycle. And the Kelvin cycle is a series of chemical reactions that allow plants in particular to fix carbon dioxide, to take carbon dioxide from the atmosphere and convert that carbon dioxide into a sugar. And the sugar is a little sugar, a small sugar. The sugar has only 3 carbons in it. It's illustrated, oops, you're going to go back the other direction. It's illustrated down here as DHAP, which stands for dihydroxyacetone phosphate. Okay, a molecule. You, Mark, stop laughing at me. Okay, here we go. I don't laugh at Mark when he preaches in my church. I don't think he should be allowed to laugh at me here. In any case, look at this figure. It looks complex, but it's simply a figure that shows a series of simple, straightforward chemical reactions. And if you were in my biochemistry class, we would talk about each one of these little reactions. But then at the end, put those reactions together and you end up with carbon fixation. You end up with taking 3 carbon dioxide molecules and producing a 3-carbon sugar. And this process has to do in 3 stages. The stages are called fixation and reduction and regeneration. This is a less favorite figure. It's not as clear. It's not as fun as that figure. Okay, let's put it that way. All right, so look what's happening here. This molecule right here, ribulose 5-phosphate, is a 5-carbon sugar. It has 5 carbons. And there are 3 of them. So each line represents 1 molecule. And so we start with these 3 5-carbon sugars right here. And those sugars are used in the fixation stage. They pick up, each one of them picks up a carbon dioxide. And if you take a 5-carbon molecule and add 1 carbon, you end up with 6 carbons, right? So you have 3 6-carbon molecules, which are then broken in half to form 6 3-carbon molecules. So now we haven't changed the number of carbons, but we changed the form of the carbons, right? This is rapidly becoming your favorite figure, isn't it? I can tell. Okay, so here we have 6 3-carbon sugars. And we move through what's called the reduction phase, change the chemistry of those 3-carbon sugars, end up here with glyceraldehyde 3-phosphate, which is an isomer of dihydroxyacetone phosphate. So we come past here, and look what's happening right here. 1 of the 6 carbon sugars, 6 3-carbon sugars, 1 goes out. 1 exits the cycle. And it's this one that kind of goes to the rest of metabolism and makes other sugars. So we can take that sugar and we can make from that sugar glucose. And that's what our corn plants have been doing up to this point. They've been capturing carbon dioxide and they've been making glucose, and then we use the glucose in a variety of ways. Sometimes we'll send it off to the ethanol plant. Sometimes we'll just pick it up in the parking lot and take it home and eat it. A variety of ways. But that all rises from the exit of this 3-carbon sugar. Right? What happens to the rest? We still have 5 3-carbon sugars, right? 5 3-carbon sugars. To complete the cycle, we need to take those 5 3-carbon sugars and convert them back into 3 5-carbon sugars, right? And that is regeneration. We're going to regenerate the 3 ribulose 5-phosphates that we began with. And all of these little reactions in between are ones that accomplish that particular end. So you take 2 of them, 2 3-carbons, and make a 6-carbon. You take a 6-carbon and a 3-carbon and make a 4-carbon and a 5-carbon. You take a 4-carbon and a 3-carbon and make a 7-carbon. You take the 7-carbon and a 3-carbon and make one type of 5-carbon and another type of 5-carbon. At the end of the day, when you go through all of those reactions, you end up regenerating the ribulose 5-phosphate, and that molecule can then capture 3 more carbon dioxides. So we have a process of fixation and reduction and then regeneration. Okay. Now, to me, that type of complexity could be considered a celebration of the physicality of creation. This is just part of the chemical kinds of mechanisms that occur within creation. This allows carbon dioxide to be captured and removed from the atmosphere in the form of sugars, right? And then, of course, we use those sugars, and when we use those sugars, we produce carbon dioxide, right? So we take those sugars, and our metabolic pattern is the kind of the opposite of the metabolic pattern of plants. We use those sugars and produce carbon dioxide. So there is a balance between the production of carbon dioxide through our metabolism and the metabolism of other animals, and the capture of carbon dioxide through the metabolism of plants. The energy for those plants, where this molecule comes from right here and where this molecule comes from right here, is what we usually think of as photosynthesis. Photosynthesis provides the power to run this particular process. Okay. So that's one example for me of the celebration of the physicality of creation. Okay. As far as I know, there aren't any psalmists that knew about that. Right? So there aren't any psalms written about that, right? But if I was a poet, I would write a psalm about that. But I'm not, so I can't, and somebody else is going to have to do that. And people that write poetry tend not to be people that enjoy molecular pathways. It's just kind of the way it is, right? Okay. A second aspect of what I do, and where I see and can celebrate the physicality of creation, is in a project that we have going on at Northwestern College. Collectively, the project is referred to as CPhages. CPhages is an acronym. It has to do with viruses. Phages are viruses, but this particular phage doesn't really refer to viruses. Although the first two are phage hunters, is the P and the H in phages. SEA refers to Science Education Alliance, and Science Education Alliance is a project of the Howard Hughes Medical Institute, of which Northwestern College is one of the schools that participates in this. So initially think, well, it's about viruses found in the sea. Well, it's not about viruses that are found in the sea. It could be, but it's really not. about those things. It's about viruses found in soil. Viruses found in soil. And so what our students do is they collect a sample of soil from wherever they want, and they bring it to the lab, and we use that soil to discover viruses, to try and find the viruses in that soil. Now, the really fascinating part of this is how many viruses people anticipate or estimate are present in the world itself, on Earth itself, right? And people estimate there are 10 to the 31st power number of viruses on Earth. Well, 10 to the first power is 10, and 10 to the second power is 100, and 10 to the third power is 1,000. So you see where I'm going there. Now take that out to 10 to the 31st, okay? There are a huge number of viruses, which means for our project is that when a student discovers a particular virus, they are the first person on Earth in the history of mankind to ever find that virus. Virus has never been found before. Now, that's a broad statement. Turns out that there are a few of them, maybe 4 or 5, that have been found before. But the ones that have been found before are on one campus, the campus of University of Maryland, Baltimore County, because so many people sample soil from that campus, right? That's the campus where this kind of originates, and their students sample, and when we go there to teach, we sample, and so, yeah, we find the same virus. They found the same virus occasionally. Okay. The more interesting case in here is that there has been at least 1 or 2 viruses that have been found in one nation and also found in another nation, and you ask yourself, how did that virus end up there and here? Which is a question that's a little harder to answer. But the fun part of this is that we discover viruses. We discover viruses. So we got to think a little bit about what viruses are. After they're discovered, their DNA is isolated, and the DNA of the virus is sequenced. And in genetics, the DNA is analyzed for what kinds of genes are present in this virus. And then in the last one, in the genes portion, Northwestern's one of only a few institutions doing the genes portion right now. In the genes portion, they take a particular virus, just one virus, and try and figure out what is the importance of each and every gene in that virus. And so the students will take a single gene and they'll analyze a single gene to see if that gene produces toxicity, whether that gene produces defense. That is a fairly involved kind of process. But we're kind of hoping most of our students that participate in the C phages will do both discovery and the annotation part. So what I'm going to describe is the discovery part. And again, think of the overarching topic here that we're going to celebrate the physicality of creation. Okay. So we're looking for viruses. What are viruses? Well, viruses are really, well, you can look at it in one of two ways. They're really either really simple living organisms, or they're really complex non-living organisms. And the question is, what's kind of your definition of life at that point? They kind of sit right at that border. I tend to think of them as living entities, Some of my colleagues think of them as non-living entities, but we still get along. Okay. But in this particular case, we're looking for a specific kind of virus, and that specific kind of virus is a bacteriophage. A phage refers to virus, bacterial refers to bacteria. These are viruses that infect bacteria. Okay. So they're relatively safe for us to use because as we discover them, they're not going to infect us. The more dangerous aspect, if there is a dangerous aspect, is the bacteria that we use, not the virus that we're discovering. Okay. So viruses, I said, are relatively simple entities. They have only two things associated with them, two molecules in terms of classification associated with them. They have a genome, and in this case, the genome is a DNA molecule, much like your genome is a DNA molecule. And they have a protein or a series of proteins that make up the body of the virus. And some of those proteins are in the capsid that contains the DNA. Some of those proteins are in tail fibers. Some of those proteins make up part of the sheath. These little individual balls here are supposed to represent individual proteins that come together to make up this sheath. So, in essence, a virus has only a genome, in this case DNA, and proteins, a variety of them, but doesn't have any of the other entities we usually associate with cells, right? It doesn't have a cell membrane, doesn't have mitochondria, doesn't have a nucleus, doesn't have anything that we associate with cells, including it has no mechanism to generate energy. It cannot make its own energy. But it replicates. And in order to replicate, it requires energy. So the energy that it uses is energy from its host cell. A virus will infect a host cell, and here you see, in today's day and age, I tend to think that whoever made this particular micrograph right here kind of photoshopped it a little bit, but maybe he was just really, really lucky and came up with this micrograph that shows 3 bacteriophages attaching to the host cell, one of which has DNA in its capsid, one of which is injecting DNA into the host cell, and one of which has already injected DNA into the host cell. If I was a good electron microscopist and I came up with this particular figure, that would make my day. That would be terrific. All right. But this is what's happening. The virus will come, attach to the host cell, and inject into the host cell its DNA. Now, the host cell is really big compared to the virus. Okay, so to give you an example of that, here's the edge, the very edge of one of your red blood cells that circulates in your blood. Okay, so think of how small that red blood cell is, and now look at this cell here. This is a bacterial cell, the kind of bacterium, one of the kinds of bacteria that are found in your gastrointestinal tract, E. coli, smaller than your eukaryotic red blood cell. And then here's a variety of viruses right here. Some of them pretty big. The Ebola virus is actually pretty large, but the polio virus is really small. Right? The polio virus here, adenovirus, infects liver cells, is relatively small. Some bacteriophages are big, and some of them are small. The ones we're concerned with are actually reasonably big. They're kind of this type of morphology. Okay. So what happens then to that virus. What does the viral life cycle look like? Well, I kind of alluded to the fact that it starts with attachment. The virus attaches to the host cell, and then the virus moves its DNA from the capsid into the host cell. We call that penetration. And once that DNA penetrates into the host, then biosynthesis begins. And the biosynthesis is the production of more DNA and the production of the proteins that make up the virus. So, what's facilitating that biosynthesis? What facilitates it is the host cell. The ribosomes of the host cell produce the proteins. And the enzymes that normally replicate DNA in your host cells replicate the DNA. And that's true in this case as well. So the host cellular mechanisms are what are captured by the virus to replicate the virus. So viruses can replicate. Viruses in the host cell can make more viruses. And so if your definition of living focuses mostly on replication, then viruses are really simple living kinds of organisms, right? If living has to do with making energy and things like that, well, they can't do that, so they're not living. You see, they kind of sit on that border. But they make DNA, and they make proteins, and then those DNA molecules and those proteins get together, they assemble, and then they release. And coming out of this host cell is a number of viruses, all identical to the first one that infected, and are able then to search for other host cells so that they can replicate as well.
Is it this 0 minute, in 2 minutes this happens, 3, 4? I mean,
Right.
all this takes place in 22 minutes?
So one cycle estimated takes place in about 22 minutes, right?
And that's true for just that one virus.
That's true for this particular virus in this particular illustration. Some viruses take longer, some viruses, I don't think very many are that much shorter than that. But it gives you a little bit of an idea of how quickly this is actually happening.
So when do we get sick from all of this? When do we start to?
We get sick when our host cells can no longer function as host cells, right? Now, it doesn't happen that our host cells necessarily die as a result of this, but for our bacteriophages, oftentimes those host cells are dying as a result of that. So what's the pathology? The pathology is the death of the host cell as a result of the viral infection. Right? And in our cases, each of our viral diseases that we think about are produced by viruses that have a specific host cell. Maybe one or maybe two, but at least there's a degree of specificity between your host cell and the virus, right? So if you take influenza infects the cells of your respiratory tract. That is the host cell for the influenza virus. Same thing holds true for the COVID virus. For the polio virus infects host cells in your gastrointestinal tract and host cells in your nervous system. So you get the effects that we usually associate with polio.